Hello, everyone. Welcome to today's webinar. I am Lini Zhang from Bristol-Myers Squibb Company, and I will be your host today. It is my pleasure to introduce today's speakers, Dr. Carrie Couton from Eli Lilly and Company, and Dr. Andreas Abend from Merck and Company. Dr. Carrie Couton is an analytical chemist with 19 years of experience in drug product development, spanning from first human dose to commercial launch. She has expertise in product performance of oral dosage forms, including dissolution and disintegration method development, analytical control strategy, and applied in silico modeling of in vitro dissolution. She is recognized for her strong contributions to the development, registration, and the launch of several products. In addition to providing technical leadership for multiple projects in Lilly's portfolio, her current projects include the development of FBRM for elucidation of tablet dissolution mechanism. She received a PhD in analytical chemistry from University of Michigan, studying programmable selectivity for high-speed gas chromatography with Professor Richard Six. Dr. Andreas Abend is a director in the Analytical Science Group in the Pharmaceutical Science Department of Merck's research labs. His group provides analytical support to early formulation development, the release and stability testing of clinical supplies, and is overall responsible to ensure new drug products entering late-stage clinical development are well characterized. In addition to his responsibilities related to analytical support of product development, he is a member of Merck's Biopharmaceutical Advisory Team, which is a resource to product development teams seeking cross-functional input towards biopharmaceutical and regulatory challenges. During his 20-year career at Merck, Andreas supported drug product and API development and provided technical support to inline products. Andreas is the co-leader of IQ Dissolution Working Group and a member of the Biopharmaceutical Technical Committee of the Product Quality Research Institute. Without further ado, let's go to Andreas.
All right. Well, thank you, Limin, and welcome everyone to today's webinar. I will go over a brief background of the presentation of today. I go over dissolution in product development and as a quality control tool. Then Carrie will talk about the CRDS roadmap, and we'll go through some examples, and then we're both taking turns throughout the process, while Carrie will then finally wrap up the presentation by talking about clinically relevant dissolution specifications and continuous manufacturing, as well as some opportunities to update current regulatory guidance. Okay, so just a brief reminder, why are we doing dissolution testing? And I find this, this paragraph that I took out of the guidance for industry for dissolution testing of immediate release solid oral dosage forms from 1997, really to the point of, again, why we are performing disso testing under various conditions for various solid oral dosage forms. And you can apply the same principles even to non-solid oral dosage forms as well, as long as the solution is really important for the drug to work. So it's stated here that the drug absorption from a solid dosage form after oral administration depends on the release of the drug substance from the drug product, the dissolution or solubilization of the drug under physiological conditions, and the permeability across the gastrointestinal tract. And because of the critical nature of the first two of these steps, in vitro dissolution testing may be relevant to the prediction of in vivo performance. And then it continues essentially that we're using disso to assess lot-to-lot quality, guide development of new formulations, and essentially to ensure continuing product quality as part of, SUPAC, scale-up and post-approval change management, and in general, for life cycle management. And this really means that we're trying to measure typically throughout the development and even in the post-approval work, the rate and the extent of drug product release. Now, what I'm showing you here is, represented by this USP type 2 dissolution apparatus. There are now 7 of these, highly prescribed or described apparatuses available for different dosage forms. And those are typically then used for quality control type testing, but not only, but also in the development space. Now, over the recent years, we've seen an emergence of either commercially available research tools like this PION dual chamber dissolution system. Companies have developed approaches which are described as, here the artificial stomach duodenum system. You have simple transfer systems here where you are exposing your solid oral dosage forms to, SGF, stimulated gastric fluids. You transfer this into another vessel which has different pH, different media composition, kind of mimic the environment of the small intestine, and so forth. And here on the bottom, I've shown you essentially a combination of a USP type 4 instrument with a biphasic system, which allows you to actually avoid supersaturation, and you can study how much drug is actually transferred into a lipid phase. But while doing all of this, we try essentially to predict the rate and the extent of drug product in vivo. Now with that, when we actually conduct our dissolution experiments in the lab, we're trying to establish an IVIVC. And very quickly here, this term IVIVC is very broad, a predictive mathematical model describing the relationship between an in vitro property of an extended release dosage form and a relevant in vivo response. So typically that's plasma drug concentration or the amount of drug absorbed. But I also like to highlight that even in this description or definition by the FDA, it says a relationship between an in vitro property. So dissolution is usually the property or the system that we interrogate. But you can easily imagine that other in vitro properties, surrogates that we often use, for example, in dissolution testing or as a surrogate for dissolution testing, can also be used to actually build a correlation to an in vivo response. So just keep that in mind when we're talking about clinically relevant drug product specifications in general. Now, what I'm showing you here is, where a company may have made 3 different tablet batches with different release profiles, tested them in vivo, and you can obtain, if you're lucky, these very nice profiles which all have different AUCs and different Cmaxes. So based on that, the level A IVIVC can be established. Now, in practice, we may be able, and you may be lucky, to actually achieve a level A IVIVC. Many times, though, for immediate release tablets and for extended release tablets, we actually see something that looks like this here, where the area under the curve, or AUC, is not dramatically different. However, you're seeing actually a distinct difference in the Cmaxes. And based on that, you can also build a correlation. Typically, what we're seeing here is called a level C IVIVC. Now, we all know that for immediate release tablets, in reality, often though, despite the fact, and we will see this in the examples that we're showing later, despite the fact that you're making different batches with different release profiles, oftentimes the PK profiles are very similar, and we cannot establish a correlation. However, this is something that's sometimes referred to as a in vitro in vivo relationship, or even a level D IVIVC. All right, so Typical applications of dissolution in the development space involves physiologically relevant dissolution testing. A lot of people refer to physiologically relevant, also biorelevant. And we're using this type of methodology to rank order formulations, typically in early development. Now, you can also use this, and companies use this oftentimes, for biopharmaceutics risk assessments, which is abbreviated here as PRA, in vivo predictive dissolution, for physiologically based absorption modeling, and you can use this to build an in vitro in vivo correlation. Most of us are very familiar with multi-pH media dissolution testing, which essentially is used, again, as part of biopharmaceutics risk assessment. It's used in biowaivers for BCS class 1 and BCS class 3 drugs, but also for biowaivers of different strengths. It's oftentimes used as part of SUPAC, for example, formulation level 2 changes for BCS class 2 containing drugs. And the intent here is to show that after certain changes or modifications, the product is still similar in vivo. The other distinctly different approach of dissolution testing is called the QC, or regulatory approved dissolution testing, which is essentially used for routine product release, stability testing. You can, again, use the methodology using traditional USP type equipment to establish IVIVCs. It's the standard required equipment for SUPAC changes. And you're using this essentially as a process performance monitoring tool. So we're essentially establishing many times manufacturing process consistency. And then there are clinically relevant dissolution specifications or clinically relevant dissolution methods. Now, I will show you a definition of the term, but all of these approaches here, physiologically relevant dissolution testing performed under multi-pH conditions, a quality control method, can have clinical relevance if you have established a link between your methods, the method conditions, and if you have a specification, the acceptance criteria, to in vivo performance. And if you have done so, you can use that method, hopefully it's a simple method, for release testing, stability testing, to support biowaivers and post-approval change management. Okay, and we kind of published this as part of the dissolution working group in 2018. And if you're interested, you can follow this link to review the paper. Now, as I said, what are clinically relevant dissolution specifications? This is an outcome of a workshop that we had in 2017 at the Maryland Center of Excellence for Regulatory Science and Innovation, where we try to achieve consistency in the use of not only the term clinically relevant, but also what are clinically, or what are dissolution safe spaces, what is biorelevant, physiologically relevant, and so on. Clinically relevant dissolution specifications are method conditions and acceptance criteria which are based on clinical performance. So that can be PK or PK/PD. And the goal is to reliably accept batches which are bioequivalent pivotal batches. A clinical safe space or bioequivalent space are the boundaries that are defined by these in vitro specifications within which a drug product or drug product batches are anticipated to be bioequivalent to one another. This verbiage is almost similar to the DA/BE guidance, where we wanted to make sure that we allow a little bit more flexibility, because typically when we demonstrate bioequivalence, we are only comparing a reference to a test drug. So that's why this addition here, or less optimally but still possible, bioequivalent to the pivotal clinical batches was added. This is very, very useful when you're trying to establish dissolution or product similarity based on your clinically relevant dissolution method as opposed to, for example, multimedia dissolution testing. Okay, I'll go over quickly some of the key elements of the clinically relevant dissolution specification roadmap that the IQ Dissolution Working Group developed in 2017. And then Carrie will discuss the roadmap in more detail. So key elements of this roadmap are an end-to-end biopharmaceutical risk assessment and risk management. This is critical to perform not only early in development, but also throughout the life cycle of the management. And I think, Maria, can you give me access back? Yeah, thank you. So you start from development, and it's really throughout the life cycle of the product. You have to always ask yourself, do I need to establish a link between in vitro and in vivo performance? That may be based on true perception of a biopharmaceutics risk, but it also may be a CMC risk, where, for example, your dissolution method may be too sensitive. Speaking of, you should really establish a fundamental understanding of the dissolution mechanism, because it's critical in identifying your critical materials attributes and critical process parameters. You need to understand these and their potential impact on bioperformance. You also want to make sure that as you develop a control strategy, that these CMAs and CPPs are under control. As I mentioned before, a lot of times in drug product development, we actually, rush through development, and we may have to do some optimization either during scale-up or even in a post-approval environment. And here you really have to think through the risk, biopharmaceutics risk, as well as CMC risk during product lifecycle management. Okay, so part of the risk assessment is also thinking through the steps that have to happen when the drug is actually absorbed. So when the drug is actually taken, what I'm showing you here is very quickly the gastrointestinal tract. Tablets usually entering the stomach under very acidic conditions, then it's transported into the small intestine where the pH now gradually increases up all the way through the passage into the colon, where your pH is typically kind of neutral, maybe slightly basic. Most of the absorption happens in the small intestine, and that is very important when we talk about gastric emptying times and gastric transit times. Now, as a drug product dissolves or enters the stomach, you may or you may not have the film coating actually dissolving, the tablet disintegrating or eroding into granules, if you have granules, disintegrating into the excipient and the API dissolving, et cetera, et cetera. The point here is that only aqueous drug, only solubilized drug can actually be transported across the epithelial cells into the portal vein and become systemic. But this also is part of understanding how quickly and how much time does the drug actually have in the GI tract. Again, this is kind of a schematic of an early development risk assessment where you start again with, trying to identify your CMAs and CPPs. You can use traditional QC type dissolution, or you can use these more, fancy novel technologies that I've shown you before, the BCS classification system, which I will talk about in a minute. Some companies still use in vivo animal models. Most of the companies are switching over into using more and more in silico models. You may have experience with the same molecule that you're developing into a new formulation, or it could be a backup formulation. But all of that kind of feeds into how you develop your in vitro methods and how you're actually identifying and controlling your CMAs and CPPs. This very briefly relates to the slide that I've shown you with the gastric emptying time and the steps that have to happen before a drug can be absorbed. For BCS class 1 compounds, these are typically considered low biopharmaceutics risk because here the only thing that matters is the gastric transit time. So in other words, if you take a drug and let's say you have a violent diarrhea and the drug just passes through, you're not really limited by dissolution per se. It's truly just the gastric emptying time. For BCS class 3 compounds, where you have low permeability, again, dissolution probably doesn't matter. You can dissolve as much as you want. If it doesn't get absorbed, then it's really not, again, a dissolution-driven process. Whereas for BCS class 2 compounds, the actual rate of dissolution may matter. We all know from first principles that both the drug solubility as well as particle size are often very critical here. For BCS class 4 compounds, you just don't know. These have typically high biopharmaceutics risk, and therefore, when you develop a method, for example, it's always critical to know how sensitive the method truly is. All right, with that, I'm handing it over to Carrie.
All right, thanks, Andreas. So this schematic shows the clinically relevant dissolution specification roadmap that was developed by the IQ Dissolution Working Group members and published in 2017. And the schematic starts with, at the top, with the sponsor decision on whether or not to perform in vivo studies where deliberate perturbations are made to either the formulation or the process in order to get different in vitro dissolution performance and then evaluate whether that performance impacts the in vivo exposure of the drug. So the two approaches are split out as 1 and 2 in the schematic. Approach 1 is really the more traditional approach where you control the formulation and the process within the range of conditions that had been established during your pivotal clinical trials and or biobatches. And so in this case, you might select this route when your process and your composition combined with your understanding of the biopharmaceutics properties will really mitigate any risk of changes in exposure for the patient. You might also select this approach in a case where conducting these additional clinical trials just to evaluate changes in the formulation might be considered unethical, either due to the indication or due to the risk of exposure to healthy subjects. Approach 2 is sort of captures all of the different variants of situation where you are doing an in vivo study to look at the impact of formulation, process, or material variants on the in vivo performance. And then depending on the type of results and the type of dose form that you're looking at, this can be further broken down into 4 different variants, which are sometimes referred to as IVIVX for in vivo, in vitro, whether it's correlation, relationship, or extrapolation. So the first variant is an IVIVC or a level A IVIVC. In this case, you've established a mathematical correlation between the drug release in vitro and the exposure in vivo. The second variant is a level C IVIVC, which is sometimes referred to as an in vivo in vitro relationship. In this case, you've established a rank order, or maybe you have established a relationship of only certain pharmacokinetic parameters with the in vitro drug release. So for example, in a lot of cases, you might have a correlation with Cmax, as Andreas illustrated earlier, but not a correlation with the area under the curve or the total exposure. The third variant is referred to as the safe space variant. In this case, you have formulation or process variants where you've intentionally changed the in vitro drug release, but when you test those variants in vivo, the bioperformance is the same, meaning the exposure is the same. And so in this case, you've established a safe space where a range of in vitro dissolution performance doesn't have an impact on the in vivo performance. And the last variant is IVIVE or in silico. And in this case, the E is for extrapolation. In this, you've established a relationship between in vivo and in vitro exposure based on physiologically based absorption models. Let's see if I can get the slide to advance. There we go. Sorry about that. All right, so first we'll go through a little bit more detail on approach 1 and an example. So in this example, we have a typical immediate release dose form where the drug product itself, the drug substance is highly soluble and highly permeable, and the drug product dissolves rapidly, so it meets the criteria for biopharmaceutics classification 1 criteria. In this case, during the development, the sponsor company found that no critical process parameters or critical manufacturing attributes had an impact on the rate of dissolution in aqueous media. And so they were able to demonstrate this during development. Also, the very rapid dissolution was observed across a wide range of process conditions. And so looking at some example dissolution data for this particular example product, the dissolution results from 7 representative batches, so rapid and consistent dissolution, indicating that this formulation and process is very robust with regard to dissolution performance. And so for this situation, default specification of either Q equals 80 or 85 could be justified based on FDA guidances that are currently in place. Or for another product, you might base this on the range of dissolution results that are observed during pivotal clinical trials in your biobatch experience. So for advantages and disadvantages, for approach 1, one advantage is it can help speed up development because you're able to avoid unnecessary and potentially costly pharmacokinetic studies. So for example, this could be justified based on the biopharmaceutics classification. But the disadvantage is that it could leave you with some questions about is your dissolution method adequately discriminating, especially if you're dealing with a drug substance that is not highly soluble. And so you could have risks on both sides of this. One would be that you're pushed to a very tight control limit and very tight process controls, which could lead to product rejects and investigations. Or you, on the other side, you have the risk that the specification that you set might be too loose and product could then be released that is not indeed equivalent in vivo. And we definitely don't want that for patients. So you would apply this approach when, based on your biopharmaceutical properties, you know that dissolution is not rate limiting for absorption. You could also apply this when you have established that the dissolution is low risk based on those properties and or a conservative control strategies in place, or in cases where additional pharmacokinetic studies would be considered unethical. So next, to go into a little more detail on approach 2. Here I show some illustrations of in vivo in vitro correlations, both type A and type C. So the type C correlation, you're able to predict your in vivo results, your in vivo absorption results directly from the in vitro. So the figure here shows that as the fraction of drug dissolved increases, the fraction of drug absorbed also increases. So this could commonly be a linear correlation that's established, but it can really be any correlation that's predictive of the in vivo performance. This type of correlation is most common for modified release formulations because of the fact that you're intentionally designing the formulation such that dissolution is the rate limiting step to absorption. It may also be possible to establish a level A correlation for a BCS2 or 4 molecule where it's not highly soluble and therefore dissolution may be the rate limiting step. A level C correlation or IVIVR is when you have a quantitative or a qualitative relationship between the dissolution and some pharmacokinetic parameter. So illustrated here are dissolution results at 15, 30, and 45 minutes, showing the correlation between the extent of drug release at each of those time points with the Cmax for exposure of this drug. And in this case, you would apply a level C correlation when variants in the in vitro dissolution impact in vivo performance, but you don't have a direct predictive correlation as you would with a level A model. All right, and so to go into a little more detail on in vivo, in vitro extrapolation or modeling, In this case, you would use mechanistic models and parameters such as particle dissolution of the drug product, permeation of the drug substance in the GI tract. You could also incorporate gastrointestinal transit rates and times, and gastrointestinal pH variance and volumes. And you can also take into account the presence of transporters, metabolism, and other pharmacokinetic parameters. And then this combined to a model can allow you to predict the bioperformance of the product, taking into account many other factors that you typically couldn't account for just in a standard dissolution experiment. So all of this together, regardless of the methodology used, the ultimate goal is to establish that dissolution range where any formulations that perform in that range or any batches that perform in that range are expected to be bioequivalent to your reference batch or your pivotal batches or BE batches. So here's an example where in vivo data was obtained and applied both in the form of an in vivo in vitro correlation type A and type C in order to predict the bioequivalent space. And you can see that the two approaches give similar results and sort of speaks to the robustness of using this approach for control. Next, Andreas, we'll walk through a few case studies for approach 2.
All right. Well, thank you, Carrie, for paving the way into the examples here for a level C IV-IVC as well as a level D or IV-IVR. So first off, we had this case published by Philippos Kessisoglu and Andre Hermans et al. In 2015 for a BCS class 2 compound with low solubility and high permeability. I'm going to flip through these slides really quick in the interest of time, but the problem here is that the formulation was sensitive towards, towards tablet hardness as well as tablet moisture. And I can't get out of this. Sorry for that. All right. All right. Yeah, the key product risk where, like I said, dissolution is highly sensitive to tablet hardness. There's a very narrow compression window. There's a high risk. Because of that small process perturbations can actually cause batch failure in terms of not meeting the specifications. And there's definitely been a significant risk for post-approval change management, as you will see on the next slide, because the F2 space in itself is very narrow. As I mentioned, the solution is sensitive to tablet hardness. And of course, we developed a method that has surfactants. There's always a question whether or not your method is truly, adequately sensitive or discriminating. So what I'm showing you here is essentially the problem that we had with this product. As you can see here along this dotted line, the response of tablet hardness to compression force is very, very steep over a pretty significant part of the compression range. When we made pivotal clinical batches, there were 2 here, they were made at about, I would say 13 to 14 kilonewton, resulting in tablets with different tablet hardness. And if you do dissolution comparison, you can see that they are still dissolution, or they would be considered similar, but this F2 space here is really not very broad. In other words, if you were to change the compression force outside of this range, you're going to be F2 dissimilar. And as we went into the clinic with these 2 batches here, we were still at the early stages of late-stage development. So the actual robust processing range was established or was desired to be somewhere in here. And in order to make sure that we have a wide enough window in our dissolution space, we actually made process variants that actually were made at extremes of compression force, both at the high end and at the low end. If you make tablets even softer, then film coating becomes a problem. If you go higher, your tablet press might stop working after a while because the stress is too high. So anyways, 4 tablet variants were made, one at target conditions here, that's formulation C, a soft tablet, and then 2 tablets which had, increase in hardness and slower dissolution profiles. And as you can see here on the right, the PK performance of these showed very similar AUC, but differences in in Cmax, I'm sorry. So we were not able to build a level A IV-IVC, but we looked into building a level C IVC. And if you look here, how you establish this is by simply walking through your dissolution rate at different time points. And I'm showing you here, if you were to take a ruler at the 10-minute time point, you can see that formulations E and D have very small amounts dissolved, around 20%. Your formulation C here is about 40–45% release, whereas the fast dissolving formulation already has achieved 100%. If you walk to the 15 minutes, to the 20 minutes, 30-minute time points, you're going to get these pictures here. And based on that, you can build a correlation between the amount dissolved at a given time point and Cmax, which is always constant because this is what we're measuring in vivo. You can use this now to then build a VE space where formulations that have a release profile that's somewhere in between here and this profile are actually bioequivalent. And that's shown here. If you were to set a dissolution spec at 10 minutes, your range would actually be 10% to about 80%. Typically, nobody does that, right? Usually, we're setting specifications around 80% release. So in that case here, rather than using this batch here, which showed full release after 15 minutes or so, almost 20 minutes, you can justify a specification of 80% release at 30 minutes. You can also use this table here to build a safe space, and that's kind of shown here. Your VE space is kind of within these blue dotted lines. And just to contrast this would be your F2 space. Now, if you're making post-approval changes and you have to do dissolution similarity, you have a much, much wider space that still shows that these changes, if the dissolution profiles happen to be outside of F2, do not impact product quality. All right. So in this case, I've just shown you very briefly here, a level C IV-IVC was established for Cmax. We have a dissolution safe space essentially for AUC. There is no signal across all the dissolution profiles shown here. And this was really enabling setting a very comfortable tablet compression for tablet hardness range, enabling then the commercial manufacturing of the product without any anticipated discards, for example. The other example I'm showing you here is something that we frequently encounter also when we're making amorphous solid dispersions, especially via spray drying. And in this case, we had to develop single entity tablets of crezeprovir for the Japanese market. This was done at a very accelerated development space. And because of that, we anticipated that during routine manufacturing, there could be some drift in the process resulting in slower dissolution profiles, which then give rise to concern in managing this in this post-approval space. In order to kind of de-risk this, we made tablets at 3 different conditions. Again, our target conditions, a very rapidly dissolving tablet, and a very slowly dissolving tablet. And you can see the dissolution profiles here. This is, again, our target dissolution profile, the fast dissolving, the slow dissolving tablets. And here we've calculated the F2s. Again, F2 of fast to target and then slow to target. They're all well below 50. And here are the PK parameters that we measured. So the geometric mean ratio of our fast tablets to the target at an AUC of almost 1. The 90% confidence interval is, between 0.92 and 1.06. The only point parameter here that for both the fast and the slow tablets would have failed the conservative PE criteria is here the 90% confidence interval. So this is a little below 0.8, and this is also a little bit below 0.8. But nevertheless, given that in this case, for this drug, Cmax is not important for safety, or the small change is not important for safety and efficacy, this was still acceptable, and the Japanese regulatory agency accepted our proposal for setting the specification. And again, this shows that this approach of demonstrating the clinical relevance of the dissolution method is not only currently accepted in the US, but also in Japan and probably in other markets as well. All right, and with that, I'm handing it over to Carrie.
All right, thanks, Andreas, for sharing those examples. I have one more example to share. This is an example of an in vitro extrapolation used to establish a safe space for lesinurad tablets. And this is a nice example from Xavier Pepin and his colleagues at AstraZeneca. The reference is shown below. The issue here was that the regulatory agency during their submission requested them to provide additional justification for their specification for both dissolution and for particle size of the drug substance. And so they were able to use existing IV and oral pharmacokinetic data to develop a model and then verify that model and apply that model to justify a specification space. All right. So for the lesinurad drug product, a discriminating dissolution method was developed by the company. The method was pretty standard conditions, 900 mil, pH 4.5 acetate buffer plus sodium lauryl sulfate. So this drug product is not meet criteria of highly soluble, and it's an immediate release tablet formulation. In this media, the solubility of lesinurad is about 4 or 5 times the concentration needed for sink conditions. And the figure here shows dissolution profiles for a couple of lots that were key to building their model. The faster dissolution profile here is representative of the clinical trial materials and was used in the absolute bioavailability study. The profiles in the middle are dissolution profiles that they expected to be bioequivalent to the faster profile. And then the third one, or the fourth one rather, at the bottom is a batch that was manufactured that had much slower dissolution performance and in the clinic was found not to be bioequivalent. And so their proposed specification was a Q80 at 30 minutes, such that they would accept batches expected to be bioequivalent and reject batches that were not bioequivalent. And so they used their modeling strategy, which is shown in the schematic here, was to develop a GastroPlus model, which was parameterized using the absolute bioavailability study, which included an IV microtracer arm. The input data from these studies were used to develop first a model to fit the IV data, and then second a model that fit the oral absorption data. Dissolution data for the various batches was then used to determine an effective particle size of the drug substance in the formulation. And this allowed them to account for any changes to the particles as a result of the formulation or the process, and gave them and the ability to predict dissolution, not only under the conditions of the quality control test, but also under physiological conditions based on that effective particle size distribution. And this gave a better fit of the predicted in vivo exposure than using either a Weibull or a Z factor due to the biphasic shape of the dissolution profile, where it's very rapid initially and then reaches a plateau. This model was validated using an RBA study, which was between the faster batch and the slower batch in the previous figure. And they were accurately able to predict the non-bioequivalence of that slower batch. So now with that validation data in hand, they used the model to predict in vivo performance of virtual batches, which were used to establish the bioequivalent space for both dissolution and drug substance particle size. And so that dissolution profile is illustrated here. You see this fifth profile that's added in between at the spec time point of 30 minutes. It's well below the proposed specification limit. And based on the modeling and the use of a virtual clinical trial, which was accounting for variability in both gastric resonance time and pH across the theoretical subject pool, they were able to demonstrate that that lower batch, that virtual batch, would be predicted to pass a bioequivalence. And so based on this, they could demonstrate that their proposed specification had a reasonable safety margin where even batches that were slightly slower would also be considered bioequivalent in vivo. And this allowed them to establish the safe space illustrated here, both for dissolution and also for particle size of the drug substance. So some general advantages and disadvantages across approach 2. Definitely advantages are you have more confidence in the level of sensitivity of your dissolution specification because your test and your criteria can be directly tied to the in vivo performance. And so this means you can assure that your commercial material performs consistently with your pivotal or bio batches. You have confidence that any batches that are not bioequivalent would be rejected by your method. And also this opens up the potential for regulatory flexibility in terms of application for biowaivers and post-approval changes. This also allows us to optimize the control space for the drug product process and formulation, because knowing what space you're in terms of whether dissolution performance is relevant in vivo allows you to reduce rigid manufacturing controls that really don't have an impact on bioperformance anyway. It also would allow you to minimize rejections and investigations due to changes in dissolution when you know you're in a space that's going to be bioequivalent for those materials. The disadvantage, of course, is doing extra clinical trials requires additional time and cost. And also, which I'll get into a little bit more detail in a couple more slides, is currently these approaches are being advocated by the FDA, European agencies, Canada, Brazil, but other countries outside of ICH, there's still sort of a question mark as to whether they're ready for implementing these types of approaches, especially for SUPAC type changes. All right, next I'll go into a few future opportunities, including application for continuous manufacturing. So clinically relevant specifications are applicable to both batch and continuous process. Well, approach 1 tends to focus more on traditional process control based on that clinically relevant dissolution spec roadmap we showed earlier. Approach number 2 really gives you both process control and assurance that your control strategy has clinical relevance. And this assures that you have high quality product delivered to patients throughout the life cycle of the product. So this is an example schematic of the development of a late stage product control strategy shared by Andre Hermans at IFPAC a few years ago. And so he's showing that early on in the development cycle, you use your risk assessments during formulation and process development to identify potential critical manufacturing attributes and process parameters. And in parallel, you're developing your discriminating dissolution method based on the properties of the product and the desired performance of the product. You then feed this into experiments where the critical manufacturing attributes and process parameters are varied and used to establish a range of dissolution response. That can then, combined with other process analytical and surrogate tests, can feed into multivariate regression models that can then be used to predict the dissolution profile based on those parameters. In a case where you have clinically relevant specifications, you've now established a direct link between your control strategy, the dissolution model, and the in vivo performance. So this is sort of the current state of affairs where you're taking your material attributes, your process analytics, and your process parameters and predicting how the dissolution is going to perform in the dissolution apparatus. And the future goal would be to leverage mechanistic modeling and absorption modeling to go directly from those process, material, and analytical parameters directly to in vivo bioavailability predictions. All right, for opportunities to improve acceptance of clinically relevant specs, first priority is really going to be global acceptance of the biopharmaceutics classification system. Currently, there are different standards across various countries in terms of how BCS is applied and granted. And there's currently an effort underway through the ICH M9, which is currently in step 3. And this will be a great boost to harmonize BCS across the ICH regions. We'd also love to see guidance and harmonization around default methods for biopharmaceutics 1 and 3 products. Currently, the FDA has a guideline that spells out their expectations very clearly, but it's not clear whether other regions would also accept the default methods that were proposed in the FDA guideline. In addition, there's room for improvement on alignment of expectations for dissolution methods for BCS 2 and 4 molecules, so those products that are not highly soluble. Currently, we see a lot of variability worldwide in terms of the expectations for both the development and application of the methods, as well as setting specifications. And this can sometimes lead to having multiple methods in place for a given batch of product in order to distribute it worldwide. And finally, guidance on how clinically relevant specs can be applied to biowaivers and SUPAC changes. Currently, those existing guidelines are a little bit out of date. They don't include any allowances for in cases where F2 can't be met, as illustrated very nicely in Andreas's example. Could we use IVIVC, IVIVR, safe space, clinically relevant specs to then justify post-approval changes in biowaivers? All right. So finally, I think we've shown in this discussion that clinically relevant specifications are those methods that establish acceptance criteria that directly relate to clinical performance. We've illustrated a range of approaches that could be used depending on the requirements and properties of the product to set clinically relevant specs. Mechanistic dissolution modeling and absorbance models allow us to establish a link between in vivo and in vitro performance that really accounts for physiological phenomena like GI transit and permeation that can't really be modeled in standard in vitro tests. And there's plenty of opportunities that exist for harmonization around the world of regulatory expectations, and continuing to influence the direction of the field by publishing and engaging with regulators in meetings and other forums will really allow us to move towards minimizing unnecessary clinical and lab testing. All right, with that, I'd like to thank the IQ Dissolution Member Working Group members who contributed to the paper and to this talk, with special thanks to Filipos and Xavier for allowing us to use their examples in our presentation. I think we can take questions.
Great. Thank you, Carrie and Andreas, for an informative webinar. So now it is time to open the floor to audience online. So I already received one question for you guys. Could you clarify in example 2 if the Japan proposal is not to perform diesel testing at release or set a spec at a later time point?
So yes, this is unclear. So for Japan, we're still performing dissolution testing at release.
Okay.
The product, it's still part of the release testing.
I see. Okay. So a second question that I have is, you have talked about the IR product and IVIVC. However, can you share the knowledge of the extended release products and the IVIVC? Do you have to establish the IVIVC for extended release product even though all the drug product batches are used in clinical trials?
So for extended release product, my understanding is that the agencies not only encourage but also expect you to develop an IVIVC. And because, like Carrie said before, because most of these extended release products are built in a way where dissolution actually matters, you should be able to A, build a correlation, and B, if you build this, I mean, I think it's a wonderful tool to guide through formulation development, through, establishing truly what your manufacturing ranges should be in QBD and so forth. So, no, I think not only is it an expectation, I would also encourage companies to do this every time.
Okay. So I have a third question, that is, how many companies perform such analysis retrospectively versus prospectively?
I'm not sure we've looked at numbers for that in terms of percentage of companies or percentage of products where this is applied. I would say it's probably a blend of either proactively identifying a specific risk of your product that may have an in vivo impact and evaluating that in vivo to confirm either way, or reactively where something happens where on stability you're seeing a change or a form change or something like that that may have an impact, which then you need to evaluate to confirm its impact.
Right.
So without numbers, my answer is it depends.
Okay. Yeah.
I would probably speculate that very few companies retrospectively do develop clinically relevant dissolution specifications after a product has been approved. The reality, maybe that's what Carrie was trying to explain, is a lot of times it's a reactionary move in late-stage development where something unexpectedly happens or you identify new risks, and then you kind of scramble last minute to establish clinical relevance of the method.
Right. Yeah. So on that, to follow up on this question, I actually have a question about how would you, what would be a holistic approach in performing the IVIVC studies? So what batch that we should use and when we should carry out the study? So can you elaborate a little bit more on that?
I think we tried to answer this question also as part of the white paper from 2017, and then there wasn't really consensus around that. But in my opinion, developing clinically relevant dissolution specification makes sense once you know what your formulation is going to look like and once you have defined the process. And I would probably, we're trying to encourage, within our own company to pursue clinical relevance of the dissolution method and specifications before going into phase 3. Because many times at this stage, your dissolution method should be finalized. And, ideally you use that method then in order to perform your scale-up and development activities, as well as generating data on registration stability and clinical pivotal batches.
Yeah, and I would
Okay.
just add that the decision to do clinically relevant in vivo testing really depends on the risks of the particular product and the design of the product. So as we mentioned earlier, if you have a modified release or extended release product where that's part of the specific design is that dissolution is going to be rate limiting, there's more of a driver to understand that performance earlier in development versus an immediate release tablet where you may expect dissolution is not rate limiting for absorption. There may not be a driver to do that study at all.
But to add to that, there may be a driver from a CMC perspective and from a life cycle management perspective. The example that we've shown you with, the BCS class 2 compound where we established the level C IVIVC kind of shows you that, yeah, there wasn't really a bioperformance risk. But if you venture into, post-approval change management, and now you have to show that two batches are similar, you have a high risk of failing F2. And in that event, what are you going to do? Do you have to run a BE study each time you're making a change, or do you want to preemptively set the specification and a dissolution safe space accordingly? So it's both, Gary. It's the bioperformance risk, but also the CMC risk.
Right, I agree. It is a balance of the risk versus the return versus the return versus investment. Okay, so I have quite a few questions coming in, so I'm going to carry on. Do you have any examples of success in the post-approval space with such models? I'll say either clinical safe space or using the clinical IVIVC approach.
Yeah, so Limen, this is Andreas here. I think those success stories are probably relatively rare, and especially with using modeling and simulation, it depends on who would accept it, what kind of data you have, and what agency is actually willing to accept those arguments. A lot of times you have success in demonstrating, for example, based on safety and efficacy or PK/PD, understanding that maybe changes in dissolution are not that important. But again, it's probably highly reviewer dependent, agency dependent, and the success rates are probably very low.
Right. Well, I think Azevia's example, right, the last case study did demonstrate that a success, by successfully build the PBPK modeling, they are able to achieve the waiver of the BE. They don't have to carry a clinical study to demonstrate the change of particle size, the input material, will impact the product in vivo performance. So the last example in this presentation did demonstrate this is a successful case for the post-approval space.
Well, I think this was at the filing stage, but also keep in mind this was for the US FDA. I don't know what the success in other markets is.
Mm-hmm. Right.
So again, I think this is the problematic that Carrie was showing at the end, that it's a handful maybe of regulatory agencies that would accept these strategies, but at the moment we don't know what the rest of the world is thinking about clinical relevance.
Okay, so I have another question. Can you give a brief account of a PBPK study to be used for the IVIVE, the extrapolation? Could you clarify for the discriminating ability of dissolution for BCS class 1 and 3 drugs? How will this related to the clinical relevant dissolution spec?
Yeah, I think for the BCS classification, where you have a BCS class 1 or 3 drug, the expectation based on those properties is that dissolution is rapid. And so the dissolution profiles that are requested as part of that classification, which consists of profiles across the physiologically relevant pH range, serve to demonstrate that independent of that physiological variability, dissolution is going to be very rapid, and therefore it's not expected to be rate limiting. And so in that case, essentially that confirmation that dissolution isn't going to be rate limiting is what makes it biorelevant. So you know that it doesn't matter as long as you're within that very rapid or rapid dissolving criteria.
Okay.
Yeah, in the most cases for BCS class 1 compounds, unless you have a very rapidly dissolving, unless you have a very short half-life, it's going to be a half-time, it's going to be, dissolution may not matter. So there are really probably a small fraction of compounds or drugs where it really matters, but in the most cases it doesn't.
Mm-hmm. Yeah.
And I think PBPK modeling can actually help answering that question.
Right, we've definitely seen examples where the PBPK modeling is used for a BCS 1 or 3 product to demonstrate, for example, that a wide range of particle sizes wouldn't have an impact on the in vivo dissolution and therefore wouldn't have an impact on the absorption. And so in that case, the modeling could then be applied to help set other specifications relevant to the performance of the product.
Right. Yeah. Okay. Great. So in one of your IVIVE example, it appears that you only had one release rate for modeling, which is not acceptable because model verification validation using different rate is crucial. So do you have, yeah.
Yeah, so in that example, they actually did have multiple rates. So the model was parameterized using an absolute bioavailability study that had both an oral arm and an IV microtracer arm. And also it was verified using a relative bioavailability study between tablet dose forms taken orally that had 2 very different release rates in vitro. So in that figure where it showed several dissolution profiles overlaid, the very fastest and the very slowest were tested as an RBA study, and then predicting the slower dissolution profiles performance was the verification arm of that model.
Great.
And yeah. I'm sort of using verification and validation interchangeably. So far, we don't have clear standards on how that's applied.
Right. Yeah, it's still working in progress.
Right, exactly.
Yeah. Okay, great. Do you have any experience working with products that are not soluble or absorbed? Does the agency have a preferred alternate IVIVC development route for compounds that do not have traditional PK data? Any?
Are they asking about like locally acting products, like something that would have local action in the GI tract, for example?
Let me see. The question is, do you have anything working for data are not soluble or absorbed? Not soluble or absorbed. I guess that would be either BCS class 4, right? Not soluble also or absorbed. That means maybe it's difficult to, yeah, that looks like BCS class 4 to me. Does the agency have a preferred alternate IVIVC development route for compounds that do not have a traditional PK? Yeah. Well, maybe it's not clear to me what the traditional PK data.
Yeah, that's why I was thinking it meant for products that are not absorbed or have local action in the GI tract. And in that case, I think your product performance testing would be focused on assuring that the drug is available for that local action. And so you might initially during development be focused on using biorelevant media, meaning media that are reflective of the GI tract or whatever site in the body. And then, yeah, in terms of the in vivo performance, I think if it's not absorbed and you're not seeing a traditional PK profile, you might have to consider other means to measure that, including you may have to consider efficacy as your endpoint rather than appearance of drug in the bloodstream.
Right.
But I don't,
agree.
I'm saying this more from a theoretical basis. I don't have personal experience in that type of area.
Right. I would imagine that if it is not absorbed through the GI tract, then the formulation should be developed in a different, let's say, parenteral formulation instead of for oral dosage form. Yeah.
Yeah, and I think if you're talking about an oral dose form where it's being enabled either to improve solubility or to increase permeability, really the principles of the clinically relevant specifications still hold, but the testing you might do might be a little different in order to assure that the product is performing the way you design and that also its effect in vivo is as intended.
I would probably add, maybe there are some biomarkers that you can measure. I could think of a product, but we haven't really established an IVIVC, not that I know of, but for example, for a cholesterol uptake inhibitor, where the action is actually in the colon, in the lower intestine, where cholesterol reabsorption actually occurs. And in that case, you could look at your lipid levels as opposed to drug absorption, but it would not be a traditional IVIVC type study.
Okay. All right. Sounds fair. If you have a high variability PK drug where the actual PK data for an IR tablet does not have the same rank order as the dissolution data, are you still able to apply the safe space approach? I think that's a question. Let me see. If you have for a high variable PK drug where the actual PK data for an IR tablet does not have the same rank order as the dissolution data, are you still able to apply the safe space approach?
Yeah, I think that definitely makes it more challenging if you have highly variable PK data. It could raise a question as to whether the lack of rank order is due to the variability or if it's due to the performance test not adequately reflecting the performance attributes that matter in vivo, if that makes sense.
Right.
So I think, yeah, it definitely would take additional consideration. And I think you would be able to look at that statistically to be able to help answer that question of is it just a matter of the in vivo data is so highly variable that we're not able to see that rank ordering. In which case, I think it would be more of a safe space approach, meaning you're looking at whether the changes to your process or formulation are impacting both the extent of absorption and the variability of absorption. And if you're not seeing a significant impact on that, then you could conclude that at least within that range, potentially you have similar exposure.
Right. Yeah. That is the definition for the safe space, right? Verify whether there is any impact from the in vitro, the manufacturing process or formulation excipient to the in vivo, performance, the PK performance.
Right. Yeah, and I know that can be especially challenging for certain indications where just because the patient population that's being studied, much higher variability may be observed just because of the patient's ability to metabolize the drug. And so in that case, you may be able to use the PBPK modeling to help understand the sources of variability and how that is impacting your ability to predict in vivo relevance of your product performance changes. So like a sensitivity analysis approach might be helpful.
Right, excellent. Yeah, okay. So for one of the case studies, the approach was approved in Japan was this same approach is successful in other markets as well?
I can answer that. This product is only offered in Japan. So we would have anticipated the approach to be, acceptable in other markets like the U.S., but yeah, it was only Japan.
Okay. All right. So in that amorphous spray dry case study, high sensitivity to compression force is likely related to formulation design. If a formulation is designed to be more robust to normal range of process variation, it helps everyone. Right, I agree. That is more of a comment rather than a question.
And one should keep in mind, with a lot of times with amorphous solid dispersions, you're going to see a little bit more of a sensitivity towards compression force. And that's maybe I'm generalizing this too much, but that's an experience that we have currently.
Yeah, I would agree with that observation. I think it's the nature of the spray dry dispersion, or that it's more compressible, and therefore it has that steeper response curve.
Right. Yeah. So the next question actually goes to Carrie. From your example on approach one, one might get the impression that it is only appropriate for BCS class 1 or 3, where the biopharmaceutical risk is understood to be low, but with an appropriately discriminating method. Couldn't this approach be used for BCS class 2 compound as well, as outlined in the recent EMA reflection paper?
Yeah, absolutely. And I think, again, it goes back to what the biopharmaceutics risks are for the product and how tight of a control strategy you're willing to put into place for manufacturing in order to ensure that all your future commercial batches of the product are performing consistently with the batches used in pivotal clinical trials or your bio batch, whatever you're using as your reference. And so you can use that approach, assuming you have an adequate understanding of the rate limiting steps of the dissolution process and which parts of the production process may then be critical for product performance, so that you can assure through other means other than that in vivo correlation that you're assuring, I think I've overused the word assuring, sorry for that, that you're making sure that your product is performing consistently with your clinical batches, but that also recognizing that you're now limited for the life cycle of that product to maintaining those production ranges and controls. So that's the trade-off.
Yeah, I agree. And actually, I have another question. This is that we have experienced with one of the product at BMS. It's also a spray dry dispersion product, and it's more of a spec setting. Do you I understand that the agencies always try to push you set up the spec. If you don't have clinical relevant data or safe space, information available, that we go the traditional route, which is the first approach for BCS class 2 compound. We have to develop a specification based on the historical dissolution data, and we have to set up a queue at instead of 30 minutes, so we have to set up a queue at 45 minutes. What would be the risk taking this kind of approach? Do you have any knowledge or experience with setting up a spec at, let's say, the queue time point at 45 minutes or 60 minutes?
So are you saying that the time point is set later based on acceptable in vivo performance or clinical performance of batches?
There is no in vivo. Let's say we do have a clinical in vivo performance was collected on the clinical batch. However, the dissolution method itself, this is go back to the question whether we should retrospectively go back to generate this kind of data. I mean, the dissolution method was developed purely based on quality control purpose, with that in mind, right? Be able to discriminate the process change or the critical material attribute change. However, we really don't have much of the experience to say we have established the IVIVC or IVIVR. We don't have those information. Then, what kind of risk that go in with proposed to the agency that we develop the dissolution specification based on our development batch, based on our, the performance of the clinical batch.
Yeah, I think sorry, go ahead, Andreas. You probably have more experience.
I mean, that's the traditional approach. I think the risk is that you have very limited batch data and, maybe just one or two, and you have to set your specifications just based on these two batches. If your process or your dissolution method is variable, your process is variable, and you're going to see that in your dissolution profiles and your, proposed Q, then that's a high risk. If you have a well-behaving product and you have really good understanding of the dissolution method, then you may be able to live with that risk.
Right. Okay.
I mean, again, I think this is still the majority of cases where companies develop, their specifications the traditional way, and they're accepting that risk.
Right. We just have to take the risk, knowing that we could be pushed back, and we have to come up with some, additional either PBPK modeling to support that a late Q is necessary, is not going to, is going to really support the clinical performance of the product. Yeah. Okay. Actually, we only have about 5 minutes left, and we still have a lot of questions. So I want to move on to give everybody a chance. So there appears to be a lack of consensus definition of clinical relevant dissolution and biorelevant dissolution test in the field. So could you clarify, or there is a need to clarify? Yes. But I think we have the paper published to describe the definition of clinical relevant dissolution and biorelevant dissolution, or so-called physiological relevant dissolution, right? In one of our IQ dissolution working group paper published. Maybe either Andreas and Carrie, you could elaborate a little bit more.
So the latest definitions were actually published by or as an outcome of the M30 workshop. And
okay.
I think that kind of tries to address the need to clarify the terminology. But even since then, new terminology has emerged, and it's very challenging. So one of the challenges for us is to really drive this consistency, not only within the US, but also into other markets.
Okay. So hopefully, someday, I mean, we will be able to come to a consensus and be able to apply that across the industry as well as globally. That is a desired state. I do agree.
Yep, and then in the meantime, I think it's just important to clarify what you're talking about. So for us, clinically relevant specs means you've got that link between in vivo and in vitro performance. whereas biorelevant or physiologically relevant is typically referring to either the media used or the use of, for example, an artificial stomach duodenum system where you're including transport and other factors.
Yeah, agree. Okay, so there's another question. Do you have experience of using TIM study data to support the clinical relevance without any in vivo to support the clinical relevance without any true in vivo? So I'm not clear. Okay. Maybe this is a question that we can address later because we have so many other questions still we want to get to. But you know, just to let everybody know that you still get the opportunity to contact our speakers today if you have any question you would like to discuss offline. So with that, let's move on to the last question I will have to say. Okay. Please specify, for example 1, which of the variants, if any, would have met the 80 to 125 requirement for Cmax? Would taking that approach end up with a tighter spec versus the multiple level C? That is Andrew's example 1, multi-level C, IVIVC example.
So can you repeat the question, Limin? I didn't.
Okay, so for your case study, which of the variants, if any, would have met the 80 to 125 requirement for Cmax? I think it's the dissolution at 1015. Is that?
So the solution curve that was shown, the second fastest, that was the target one. So if you look at VE criteria of the fastest versus, you know, the target and the slowest, these were all non-equivalent based on Cmax to the target.
Okay. And would it take that approach end up with a tighter spec?
No, not really, because what you've seen is essentially that we had a much wider specification. The slower profiles that were then used to establish the VE bounds were still giving you, I mean, that was shown on this one plot where you could have picked any of the time points on this dashed blue line to set your specifications. However, you know, our experience is that the regulators typically look for a Q of 80, and they usually push, you know, somewhat into the BE space so that while you not may get the entire benefit of using the slowest batch, you're at least getting a substantial, let's say, room to maneuver in.
Yeah, establishing a safety margin.
Yeah.
Okay. Yeah. All right. Thank you, Andreas, and thank you, Carrie, for you know, a very interactive session with the audience. We still have a few more questions. Actually, we didn't get to, but again, I'm going to give the floor to Maha Leo, and she's going to wrap up the session.
Sure. Once again, thank you very much for joining us, and I hope you have a great afternoon.
Yeah, thank you, everyone.
Thanks.